Namaste, dear student. Today we discuss about the mechanism of action of antibiotics. Here you can see in this slide we discuss about the general mechanism of whole antibiotics. First one, the cell wall synthesis inhibitor. Here you can see the beta-lactam antibiotics are the main group. Those inhibit the cell wall synthesis. In this include the penicillin, cephalosporin, carbapenem, and monobactams. Other varieties of the class that also inhibit the cell wall synthesis inhibitor, vancomycin and bacitracins. And another drug group that also inhibit the cell membrane inhibitor. Here you can see inside the membrane we can see this is the cell membrane, and outside the blue coloration membrane here you can see those membrane we can say this is the cell wall. Cell membrane is inhibited by the polymyxin. So this is the three varieties of the class. Those are mainly used for cell wall synthesis inhibitor. Second main group, this is the nucleic acid synthesis inhibitor. First one in this, the folate synthesis inhibitor. Second group in this, DNA gyrase inhibitor. And third one, this is the RNA polymerase inhibitor. In this first one, folate synthase inhibitor, these are the drug, mainly sulfonamide and trimethoprim. That means sulfonamide is alone also, and trimethoprim combination with sulfonamide, this we can say this is the cotrimoxazole that also inhibit the folate synthesis. That means here you can see PABA, para-aminobenzoic acid, those are converted into dihydrofolate reductase. This dihydrofolate converted into tetrahydrofolic acid. This sulfonamide that inhibit the PABA to dihydrofolate in the presence of dihydropterol synthase enzyme. So this drug that inhibits the dihydropterol synthase enzyme, and trimethoprim that inhibit the dihydrofolate reductase enzyme. These are the responsible for synthesis of the RNA as well as the proteins. These are most important material for development of bacteria. Same as the DNA gyrase. Main example is the quinolones. These enzymes are responsible for replications of DNA. If the replication are happening, multiplication of DNA are happening, and multiplication of DNA are happening, this is the responsible for development of bacteria. So this quinolone that are inhibit the DNA gyrase, and automatically nucleic acid synthesis are inhibitor. Same as the RNA polymerase are also responsible for multiplication of bacteria. So rifampicin that inhibit the RNA polymerase enzyme, and multiplication cannot be happening. Third varieties of the mechanism of action from the bacteria are it is the protein synthesis inhibitor. The protein synthesis is the most material for development of the bacteria. In these protein synthesis are specifically happening in the ribosomes. Ribosome, it has a 50S ribosome and 30S ribosome. The sum of the drug that inhibit the 30S at the synthesis of the protein at 30S sites, specifically tetracyclines and aminoglycoside. Aminoglycoside include the gentamicin. It's the best examples of aminoglycoside, and streptomycin is the best example of aminoglycoside antibiotic that are inhibited this particular 30S ribosome sites. And another varieties of the drug that inhibit the 50th subunit site, example is the macrolides, clindamycin, linezolid, chloramphenicol and streptogramins. So this is the basic mechanism of antibiotic where that inhibit the growth of bacteria. Thank you very much for watching PharmaJosh YouTube channel.